Hi student, I am Dr. V.S. Thavre. Firstly, I share the screen of today's lecture in front of you. Welcome to the fifth lecture of course of fifth semester, Path 354, Diseases of Field and Horticultural Crops and their Management. In today's lecture, we discuss smut disease of field crops. So, in sorghum, four types of the smut disease are seen. That is, grain smut caused by Sporosporium sorgi or Sphaerotheca sorgi. Second one, long smut, Taliposporium ehrenbergi. Loose smut, Sporosporium cruentum or Sphaerotheca cruenta. And last one, head smut, Sporosporium rilleanum or Sphaerotheca rilleana. So this is the four type of the smut seen in sorghum, and in rice, fall smut caused by Ustilaginoides variants or Claviceps oryzae sativa. Okay, so firstly, we discuss the four types of the smut seen in sorghum. So today we discuss these four type of the smut in comparatively manner. So first symptom point. So in grain smut, this grain smut is also known as the covered smut or kernel smut or short grain smut. So this type of the infection seen on sorghum at the time of the grain filling or grain formation in the infected airheads. So after infection, the individual grain are totally replaced by smut sori. So this smut sori generally same means size of the infected grain and size of the healthy one grain is same. So these infected grains are generally tough, means these smut spores is unbroken until threshing operations, or these spores broken due to the mechanical pressure, so release the black spore mass. Means these infected grains having creamy white in color with tough walls, so this is known as the peridiums. Okay, so this grain is crushed with your fingers, so release the black powdery mass. So this is the symptoms of grain smut. Then in regarding the long smut, in long smut disease, only few grains are converted into large smut sori, and these large smut sori scattered irregularly within these infected airheads. Okay, so these infected grains or sorus is covered by whitish to dull yellow thick membrane, and this infected grain is much larger than this grain smut disease. Means this infected grain is always larger than healthy one grains. So this grain infected grains is always cylindrical in shape, and this grain is remain intact until broken. Suppose the mechanical pressure is developed, so release the black powdery mass. So this is the symptoms of long smut. Okay. Then we go to the remaining two diseases, that is a loose smut and head smut. So in regarding the loose smut, so this is the healthy one airhead and this is the infected airheads. Means the infected plants are stunted in appearance and produces thinner stalk. Also more tillers is developed or produced, and this infected plant produces earlier flowering than the normal plant. So this is the indication of the loose smut symptoms. So in these symptoms, all the spikelet of an infected airhead or sometimes only few spikelet in infected airhead are converted or malformed and total hypertrophed in nature. Means the sorus is replaced by pistils, anthers and stamens. So the ovaries is totally converted into large smut sori. So these infected airhead appears or looks like as a leafy or leathery structures. Means infected spikelet produces the short leafy structure. So this is the also indications seen in the infected airheads. Means in these infected airheads, the infected spikelets having the black powdery mass, and these black powdery mass are easily released through these infected airheads. Then last type of the smut, that is head smut. So the infected airheads is not produced in the infected plants. Means this type of the structure generally produced or emerge from the boot leaf stage of the sorghum plants. Okay, means normal inflorescence are replaced by sorous membrane, and this sorous membrane is fully covered by greenish white membrane. So this type of the structure is released at the boot leaf stage of the sorghums. Means the pathogen generally infect at the time of the flowering season of the sorghums. So this reproductive organs is totally converted into large fibrous or filamentous bodies. Means the sorus fully emerged fungal wall rupturing or exposing large mass of the black powdery mass. Suppose you see the single filamentous. So this single filamentous generally long, thin, dark colored. So looks like as a hairs. So therefore this head smut symptom is locally known as the Jipari structure or Kesal Kanni. Okay, means in this case the entire airhead or totally airhead is transformed into long hair-like filamentous bodies having brown to black spores are developed. So that's why this disease is known as the Kesal-Kani. So this is the major difference regarding the symptoms in these four type of the smut seen in the sorghums. Okay. Now we go to the next point, that is the etiology. Etiology means morphological and taxonomical characters of particularly pathogens. Okay, so see the disease cycle or life cycle of these Phasolotheca pathogens or Sporosporium pathogens. Okay, means suppose the infected grains, or at the time of the harvesting, some of the infected grains fallen on the lands or soil. So at the time of the plowing operations, these smut spores or these infected grains debris in the soil, means actual these spores is developed dikaryon mycelium. Means this dikaryon mycelium become persist for a long life or long duration in soil. After favorable conditions, this dikaryon mycelium germinate and to produce the chlamydospores. So these chlamydospores germinate under the favorable conditions and to produce the four-celled promycelium. Okay, so this is the 1, 2, 3 and 4. So this is the four-cell promycelium is produced after the germination of the chlamydospores. So this four-celled promycelium is also known as the basidium. So this basidium produces the four type of the basidiospores. So these basidiospores produced intercalary or terminally to this basidium. So this basidiospore is also known as the sporidia. So this sporidia is released from this infected grain or infected basidium and directly fallen on this airhead. So the infected airhead having the grains, so these some grains are converted into smut sori. Means the primary infection, this is primary mycelium and secondary mycelium infect to the developing grains. So these developing grains are totally converted into smut sori, means actually black spore mass is produced. So this is the life cycle or disease cycle of the Sporosporium or Phasolotheca pathogens. Okay, so in grain smut, this is the infected sorus, means these healthy one grains become infected and produces the sorus. So this sorus having the columella is present. Okay, so you just see this infected grains. So this infected grain or inside the peridium, large columella is present. So this is known as the columella. So columella is always present in grain smut pathogens. Okay, so this infected grain having the spores are germinate under the favorable conditions and to produce the four-celled promycelia. This is the 1, 2, 3, and 4. So this is the 4-celled promycelium produces the basidospores or sporidia. So this sporidia is released from this basidium and directly deposit on the healthy one hair heads or infect to the developing the grain. So these developing grains become infected into smut sora. So this is the smut spores means sporidia. So this sporidia is always oval to conical in shape. Then long smut. So this is the infected airhead in which some grains are infected. So this is the smut sorus. So this smut sorus rupture and release the black powdery mass. Means actually they germinate the sporidia. Means actually this sporidia is produced after the germination of the teleospores. Okay, so this is the actual spore germination will be takes place in also regarding the long smut. Okay, then loose smut. Okay, so this is the infected airhead of these sorghum plants. So this infected airhead having the smut spores, so these smut spores easily blown and fallen on the jawar plants at the time of the flowering. Means these spores directly deposit on the flowers. So these flowers means spikelet is totally converted into smut sora. Means these infected flowers cannot develop the seeds. Means the flowers, infected flowers having the smut spores are produced. So this infected airhead in which the some portion of the airhead to be converted and remaining portion to be healthy one. So this healthy one portion of the airhead having the seeds. So this blackish core mass generally stick on this healthy one seed. So this healthy one seed become infected means this mycelia become persist on the outer side of these jawar seeds. So such infected seeds so in the main fields means these mycelia persist or become systemic in these germinating seedlings but not develop the symptoms on the leaf portion. So at the time of the airhead formation or after the airhead formation, these spores directly infect to the developing grains and these developing grains or these developing floral parts are totally converted into loose black spore mass. So this is the disease cycle of the loose smut. Then head smut. So same that is sporidia generally produced after the germination of the chlamydospores or that is the teleospores. Okay, then disease cycle of this head smut, loose smut and grain smut. Okay, means this grain smut, loose smut and head smut is a seed-borne infections. Okay, means the different type of the symptom is produced on the airhead of the sorghums. But you see the individual airheads. So on the airhead, only few spikelet or only few grains are converted into smut sora, but remaining grains become healthy ones. So at the time of the threshing operations, these infected grains having the peridium structure, so they release the blackish powdery mass from the peridium wall of the infected grain, and this mass generally mixed or this mass generally stick or added on the surface of the healthy one grains. So these healthy one grains become infected, and this grain, infected grains, generally used for sown in the main field in the next seasons. So this mycelia become germinate along with the germination of the seed. So entry of this mycelia takes place through mesocotyle and hypocotyle, means pathogen develop teleospores or chlamydospores in the winter stage of this fungus and finally develop the sporidia. Okay, means suppose these pathogen survival in this seed or soil as a teleospores. So basidiospore generally form. So these basidiospores means sporidia is developed by dichrotic mycelium, and this dichrotic mycelium is infect to the young seedlings of the sorghum. So these young seedlings develop the airheads, means these basidiospores infect to the developing grains. So these developing grains become infected and develop the smut spores, means blackish spore mass is developed inside these infected grains. Means actually teleospores are produced, means sporidia is produced. Okay, so at the time of the threshing operation, this pathogen or this blackish spore mass having the spores mixed with the other seeds during the harvesting or threshing operations. So again basidiospores is produced. So in this way, the complete life cycle of the grain smut, loose smut and head smut pathogens. Okay, then we discuss regarding the long smut or disease cycle of the long smut seen in the sorghum, because long smut is a airborne disease, means the actual blackish spore mass is developed in infected airheads or infected grains. So these grains may be debris in the soil. Means actually teleospore survival in soil for a number of days. After favorable condition, these teleospores germinate and to produce the basidium. So at the tip of the basidium, 4-celled promycelia is produced. Means finally basidiospores are produced, and these basidiospores directly fallen on the developing airheads having the flowers. So these ovary get infected by these basidiospores. So these basidiospores mainly convert the ovaries into larger smut sora. So finally develop the symptoms in airheads. Okay, means the primary infection generally takes place in grain smut, loose smut, and head smut takes place through the use of the infected seeds. But in regarding the lung smut, this is the airborne disease. Okay. So now we comparatively study the information of these four type of this smut in sorghum in very short manner. Importance, that is this grain smut is the most destructive in nature and common seen in the all the type of the variety of this sorghum. But long smut is a less destructive as compared to the grain smut. Means the remaining three disease is less common, or this infection generally seen up to the 10%. So this is the less destructive as compared to the grain smut. Okay. Then causal organism, Sporosporium sorgi, Sporosporium purentum, Polyposporium ehrenbergii, and Sporosporium lilianum. Then air infections, means in grain smut, only few grains or each grains are converted into smut sora. So this condition also be seen in long smut. But in loose smut, all the florets or all the grains are converted into smut sora. And in regarding the head smut, totally airhead is converted into longivary-like structure. Then site of infection means fungus or pathogen infect to the ovaries in regarding the grain smut, long smut, and loose smut. But in regarding the head smut, this pathogen infect to the flowers or to the inflorescence. Then in regarding the size of the sori, generally small type of the sori seen in grain smut and long smut, but long sori generally seen in loose smut, and very large type of the sori seen in the head smut. Okay, then columella. So columella is present in infected grain in regarding the grain smut and long smut, but absent in loose smut and head smut. But in loose smut and head smut, vascular strands or vascular filamentous structure may be produced in absence of the columella. Okay, then serous membrane. So this serous membrane generally tough and persist in grain smut, thin and rupture easily in loose smut, tough and persist in long smut, and thin and rupture easily in head smut. Okay, then the spores. So in regarding the grain smut, single round to oval, smooth walled, olive brown infected grains are seen in grain smut. In loose smut, single spherical or elliptical size of the spores, generally dark brown in colors. Then in regarding long smut, angular to globules. So these spores generally unite in spore balls and brown in color, and head smut loosely bound into balls. So these spores always spherical and dull brown in color. Then viability of these spores means survival of these spores in the absence of the main host crop under unfavorable conditions. So in regarding the grain smut, this type of the pathogens its survival over 10 years or above than 10 years. In loose mud, these spores viable for 4 years and remaining two type of the disease, that is lung smut and head smut. In this type of the disease, this pathogen survival about 2 years. So this is the viability of the spores. Then actual primary source of infections means internally seed-borne or infected seeds in regarding the grain smut, in regarding the loose smut and head smut. But in regarding the long smut, the sporidia released from the infected seeds. So this sporidia acts as a primary source of infection. Then secondary source of infections means all the 4 type of the smut disease. Generally sporidia is produced and this sporidia released through wind means sporidia generally released from the infected airheads and deposit on the another healthy one airheads through wind or air currents. So this is the general characters in different points of the grain smut, loose smut, long smut and head smut. Okay. So this is the basic information about symptom, causal organism, etiology, disease cycle and perpetuation. Then last points, that is a control measures. Means how to control this whole type of the smut seen in sorghum. So first points to be considered, that is a use disease-free seed, because the generally primary infection takes place through the use of the infected seeds. So these seeds generally treated with fine sulfur powder at the rate of 5 gram per kg or organomercurial compounds at the rate of 2 to 3 gram per kg of seed. So finally to control the disease effectively. Okay, then follow crop rotation with non-host crop, because this pathogen becomes survival in soil or in seed is 10 years, means 2 years up to the 10 years. So it is necessary, do not cultivate the sorghum every year in the same field year after years. Okay. Or suppose already disease is produced and develop the symptoms on airheads. So collect these airheads. Do not broken the infected seeds. So collect these smutted airheads in gunny bags or cloth bags. Means just cover these gunny bags from the top portion of the airheads up to the end points of the airheads. Then these gunny bags is tied with the help of the strings and then cut the airheads. Means cut the airhead below the infection points by covering the gunny bags over the airheads. So these gunny bags dip in boiling water or boiling pan having temperature is near about 40 to 50 degrees Celsius. So in this way, kill the blackish spores, that is sporidia. Then last points to be considered to eradicate, avoid the grain smut disease in sorghum by growing the resistant variety, that is a PJ 7K, PJ 23K, the Nandial. So this is the several cultivars or variety is somewhat resist to moderately resist against the grain smut disease. Okay. Then regarding the long smut, the early sowing of the jowar seeds may escape the disease by non-synchronizing the flowering with the sporidia release. Then another practices is same as regarding the grain smut disease. Then loose smut, this management is also same, already told to you. Then head smut, okay. So this is the management points to be considered to avoid the infection of the head smut pathogen to the sorghum plants and produces the head smut disease. Okay, so this is the basic informations regarding the grain smut, loose smut, long smut, and head smut. Okay, now we discuss the most important disease seen in rice, that is a false smut. So this is the most important disease seen in rice. So this is the infected rice plant in which the infected panicles or infected spikelets having some yellow color spore balls are seen, or some grains means some blackish spore balls are seen. So this type of the infection generally seen in rice plant. So this disease is known as the false smut disease. Okay, so you see the infected panicle or infected spikelets of the rice plant. So this infected grain is totally transformed into large smut sore. Okay, means these pathogens infect to the young ovaries of the individual spikelets and transformed into large smut balls. So initially these smut balls are generally yellow or somewhat orange in colors, and these orange smut balls covered by a membrane. So only few spikelets in a panicles are generally infected, and these infected smut balls are seen on the rice plants. Okay, so after the heavy infection, these orange or yellow membrane spore balls rupture or burst. So this yellow color or orange color looks like as a olive green in colors. Means this is the initial infection of the pathogen. These infected smut spores are generally yellow in color. But at the middle stage of the infection, these infected smut balls generally olive green in colors. And in the last stage of the infection, these yellow smut spores is totally transformed into blackish smut spores. Okay, means Only few grains are infected in a panicle, and these grains develop into smut spores. Okay, so this type of the infection generally seen in rice plant due to the Mucilaginoides varians pathogens. So this type of the pathogens develop the symptoms after the flowering seen in the rice. Okay, so this is the symptom of the Halsmer disease in rice. Okay, means in last stage of the infection, the round to oval sclerotia generally seen on the infected panicles. So generally these sclerotia generally hardy, compact, black in color than the normal grains. So this sclerotia having the size that is a 10 to 12 mm in size. Okay, then go to the next point, that is a causal organism. Imperfect or conidial stage, that is Mucilaginoides varians. Means this pathogen reproduced by sexually as well as asexually. Means in this disease, the sexual spores and asexual spores are produced. So sexual spores generally produced in Claviceps oryzae sativa, that is ascospores is reduced. And in regarding the Mucilaginoides varians, the Sporidia or chlamydospores are reduced. Okay, so in Mucilaginoides varians, generally isolate in laboratories. So creamy white cottony growth seen. So this mycelia generally fluffy, compact and leathery in natures. Okay, so this mycelia develop under favorable conditions and to produce the chlamydospores. So these chlamydospores is always spherical to elliptical and olive green in color. Okay, means these chlamydospores germinate under the favorable conditions. So develop the spores, and these spores is known as the sporidia. So this sporidia generally infect to the healthy one spikelets in a panicle of a rice plants. Okay, then this fungus also be reproduced by sexual methods. Means this sclerotia germinate under the favorable conditions or germinate from the infected stoma. So these stoma having the sexual spore fruits are produced, and these sexual spore fruits is always flask set containing the asci and ascospores are produced. So this is the asci and ascospores is produced from this sexual Pailthecium fruiting bodies. Means primary infection is always takes place through ascospores released from this perithecia and infect to the healthy one panicle of a rice plant. And secondary infection means already blackish spore fruits having the sclerotia is produced. So this sclerotia acts as a secondary infection. So spread of the disease from infected rice plant to the healthy one rice plants through sclerotia by various agencies. Sometimes this pathogen is also survival in different alternate host, that is barnyard grass, Congon grass, or some wild variety of the rice. Okay, so these pathogens produces the toxin, that is ustilagotoxin. So this toxin produced by these Ustilaginodine pathogens so easily penetrate this pathogen in the healthy one grain of the rice plants. Okay, so this is the disease cycle of the false smut in rice. So this is the asexual cycle, means sclerotia is produced, and in sexual cycle generally ascospores and asci is produced. Okay, and last, that is a control measures, means how to control this false smut disease in rice, because drastically reduce the yield of the rice due to the heavy infection of the false mud seen in the rice. So avoid flow of water or irrigation water from infected field to the healthy field, because this sclerotia can disseminate through waters. Then early planted crops has less mud balls than the late planted crop. This is another effective management to avoid such type of the infection in rice plants. So at the time of the harvesting, diseased plant should be removed and destroyed. Means sclerotia do not fall in the fields. Means to reduce the primary inoculum for the next years. Then these pathogens also be survival on the collateral host. So you must be taken the method, that is eradicate the collateral host present in bunds of a rice fields by applying the some weedicides. Then excess application of nitrogen fertilizer should be avoided. Means you can apply the proper or recommended dose of the fertilizers, that is NPK, to particular rice crops. Then seed treatment with carbendazim at the rate of 2 gram per kg of seed, because the infected airhead having only few spikelet are converted into smut balls, means remaining grain is healthy one grains. So this healthy one grain is also infected. So such infected grain is used for sowing purpose. So 100% disease is produced. Means to protect the seed from the fungal infection by treating with a fungicide, that is a carbendazim 2 gram per kg of seed. Suppose already infection is developed in rice plant. So immediately this rice field spraying with the copper oxychloride at the rate of 2.5 gram per liter or propiconazole at the rate of 1 ml per liter at wood leaf stage and milky stage. So this is the stage of the this is the two growth stage of the rice. Means initial infection generally seen in rice plant at wood leaf stage, but symptoms is not developed. But after the development of milky stage, this fungus attack to the ovaries. So these ovaries develop into smut sooner. Means to protect the crop from the fungal infection at wood leaf and milky stage by applying copper oxychloride and propicanozoles. And last management, that is a growing resistant variety IR22, IR26, IR28, Surya and Vijaya. So this is the resistant variety grown by the farmers to avoid such type of the infection in rice. Okay, so this is the information about the false mud disease in rice. Okay, any doubt in today's lecture? If any doubt, you can call to the mail or you can call to the me as a personal by using SMS or just mobiles. Thank you.